Okay, let's start. I am Pharmaomics. Today's lecture, I will tell you about the protein groups, major protein IDs, leading protein, and unique major peptide. Before we go deep dive into today's topic, I will explain the bottom-up proteomics, which is based on today's topic. Bottom-up proteomics uses peptide information to identify and quantify proteins. First, proteins were extracted from our biological samples. Sample preparation breaks down protein samples into smaller peptides using enzymes, such as trypsin. This peptide is then separated by liquid chromatography and introduced into a mass spectrometer, which detects them based on their mass-to-charge ratio. After that, raw data from mass spectrometry was processed using Maxquant to get protein identification and quantification data. Usually, the commonly used file format is a text file which can be opened using Excel. Important point is that we can get protein data from peptide content. Peptide is very important to know which protein is present in our sample. Note that proteins will be denoted with uppercase English letters and peptides with lowercase English letters. Okay, now we are ready to understand our topic. Let's deep dive into today's topic. To understand protein groups, all you need is to know the concept of sets that you learned in high school. There are 4 proteins. Peptide X, Y, Z were associated with protein A. Peptide X, Y were associated with protein B. Peptide X were associated with protein C. And peptide Y, P were associated with protein D. Okay, from now on, we assume that protein is a set and peptide are element. Is protein B a subset of protein A? Yes. Is protein C a subset of protein B and A? Yes. So, protein A, B, C contained in the protein group. Is protein D subset of protein A or B or C? No, it cannot be a subset because it contains a peptide D which is not present, a peptide P which is not present in the other proteins. So protein D doesn't contain the above protein group. It's easy, right? From now on, protein group 1 include protein A, protein B, protein C, and protein group 2 include protein D. Okay. Okay, let's see unique peptide and racer peptide. Unique peptide should belong to one protein group, and racer peptide is shared with other protein groups. We have 2 protein groups, right? As peptide X and Z is present in only protein group 1, protein A, B, C, not other protein group. We call it unique peptide. However, peptide Y is also included in not only protein A and protein B in the protein group 1, but also protein D in protein group 2. So we call peptide Y racer peptide. Peptide X, Z is unique peptide, and peptide Y is a racer peptide. Okay. Our last topic is leading protein and major protein IDs. Leading protein is the one with the most peptides. Our slide protein A has peptides, 3 peptides, which are biggest number among protein A, B, C. So protein A is the leading protein. It's easy. And what is major protein IDs? Major protein IDs should have at least half of the peptides that our leading protein has. Okay, in our case, how many protein A has? 3, right? And what is half of 3? 1.5, right? So if protein in the protein group 1 So if protein in the protein group 1 have more than 1.5 protein peptides, it can be a major protein IDs. And in our case, protein A and protein B are the major protein IDs. Okay, this is real Maxquant output file. There are many columns in this file. First column, protein IDs show every protein in the protein groups. Each row contains each protein group information. Look at 180 rows. There are 4 proteins belonging to one protein group. These proteins are separated by semicolon. Let's look at the third column before we look at the second column. There are 4 numbers 9, 6, 6, 3 separated by semicolon. So it means first protein O00534 have 9 peptides, and second protein O00534-3 has 6 peptides. And what is leading protein in these protein groups? Yes, first protein O00534, which have 9 peptides. Okay, now we are ready to see second column. And second column showed major protein IDs. What is major protein ID? As you saw in the previous slide, major protein IDs are that have at least half of the peptides that our leading protein has. In 180 row, O00534, which have 9 peptides, is leading protein. So what is half of 9? 4.5. Yes. So proteins having more than 4.5 peptides can be major protein. So in this case, O00534, O00534-3, O00534-2 are major protein IDs. Great, it's done. Today's lecture is done. Thank you for watching.